Hi, my name is Volodymyr Krotov and I am going to present to you our findings that have been recently published in Pain.
Lamina 10 is the gray matter area surrounding the spinal central canal. The neurons residing in this lamina play important roles in a number of processes, including control of motor neuron function, autonomic regulation, somatosensory integration, and visceral nociception. Yet, lamina 10 remains poorly studied, and the mechanisms of information processing in this spinal cord region are still unrevealed. To study the neurons around the central canal, we took advantage of spinal cord hemisection that created an immediate access to the lamina 10. This procedure preserved the entire rostrocaudal extension of the dendritic tree of the neurons, their ipsilateral primary afferent input, and the local neuronal network. Using oblique LED illumination ideally suited for cell visualization in thick blocks of tissue, we performed visually guided patch clamp recordings coupled with electrical stimulation of the dorsal root. Such experimental approach allowed investigating the intrinsic properties, primary afferent inputs, and discharges of the lamina 10 neurons. Though being quite diverse in terms of their intrinsic properties, lamina 10 neurons showed many features common to the pain processing neurons from the superficial dorsal horn. Lamina 10 neurons exhibited tonic, adapting, single spike, and delayed firing patterns. Approximately 1/10 of cells showed bursting pattern, which in the superficial lamina 1 is related to a subclass of anterolateral tract projection neurons. Lamina 10 neurons are directly supplied by 4 classes of thin afferent fibers. Based on their conduction velocities and electrical activation thresholds, these afferents belong to A delta, high threshold A delta, low threshold C, and C types. The dorsal root stimulation also evoked the polysynaptic excitatory component, which increased the overall efficacy of the input. We did not reveal any mono or polysynaptic inputs mediated by the low threshold A beta or A delta afferents, and therefore it is likely that lamina 10 neurons are preferentially involved in processing of sensory information from the high threshold nociceptive afferents. That correlates with an important role of lamina 10 in the processing of painful signals. Apart from excitatory inputs, a half of lamina 10 cells also received polysynaptic inhibitory inputs. Inhibitory currents were elicited by the activation of high threshold A delta and C type afferents and usually followed the initial excitatory input. Such robust inhibitory control might function as an afferent and/or network-driven negative feedback, terminating induced depolarization and limiting the number of action potentials evoked by the peripheral stimuli. To prove primary afferent inputs are functionally significant in lamina 10 neurons, we examined their afferent-driven firing. The A delta fiber range stimulation of the dorsal root induced a spike in only 8% of neurons. This was in agreement with the results of our whole cell experiments, showing few inputs from A delta and low threshold C afferent fibers to lamina 10 neurons. However, the root stimulation at intensities activating high threshold afferents evoked spikes in 1/3 of the neurons tested. In most cases, the response consisted of one spike, which was triggered by the long latency excitatory postsynaptic potential components, mediated by C fibers. Thus, lamina 10 neurons are active elements of the intrinsic spinal network that are directly targeted by nociceptive afferent fibers and process diverse modalities of peripheral input. The differences in the primary afferent inputs evoked and spontaneous discharges seen in the neurons with distinct intrinsic firing properties are likely to reflect an involvement of specific populations of lamina 10 neurons in diverse signal processing circuitries.
Thank you for your attention.